Hello, welcome. We'll give just another minute or so for those who are still logging on. Happy Tuesday to all of you out there. Perhaps a few of you are in a part of the world that has rolled into Wednesday. Welcome everyone to today's webinar, Digitizing and Automating Clinical Pathology in Your Lab, sponsored by TechSite. Troy Bankhead and I are delighted to join you. Troy and I are both part of the go-to-market team here for TechSite. I sit in the Salt Lake City, Utah market of the United States. Troy sits in Luxembourg in Europe. Today, we'd like to cover a few things. First, we'll introduce who TechSite is. We'll describe why laboratories are going digital, why they should consider going digital. And we'll share 3 steps that we've seen and observed as laboratories have made the transition from traditional microscopy to various methods of digital microscopy, from digital slide management only to digital slide reading, all the way to the ultimate of computer-aided slide reading for traditional microscopy-based tasks in the lab. We'll also hear from Troy about the process and change management laboratories should be thinking and planning ahead for, and potential economic and performance metrics impacts as they consider automating and digitizing their clinical pathology in the lab. And as always, we'll have plenty of time for Q&A. I should also mention that feel free to send questions via the chat. We would love to have your input throughout the session, and our moderators will be looking at those, and Troy and I will do our best to answer your questions. A bit about TechSite. TechSite has been around about a decade. We were originally a technology transfer out of the University of Utah and ARUP laboratories. We have grown big and fast, and we're now over 120 team members, head count. We are fully remote, but we do have dual headquarters in Luxembourg and the Salt Lake City area. Of course, today we're focused on our human vertical, our medical vertical, but we have developed software that has been deployed in over 5,000 clinics globally in veterinary, as well as a mold and air particulate software product for environmental home inspectors and other laboratories that are looking for the presence of mold. When we talk about TechSite being a clinical pathology AI platform, we are more than just an AI software company. We recognize that AI is great, but for a laboratory, without it fitting into a laboratory's existing workflow, AI really falls flat. We've built a platform with our software that everything from sample prep to scanning that sample, getting a good image, ingesting that image in a secure, scalable environment that's compliant to regulations, that's HIPAA compliant, and that allows labs to take the results that are populated from the TechSite AI, import those results into their LAS for reporting, all of that within a robust image management system and IMS coupled with AI to help digitize and automate and create efficiencies for a lab. So when we talk about platform, not just referring to the AI itself, the algorithms, but everything around that that can actually help a lab to drive accuracies and efficiencies. On the left-hand side of this slide here, those are some various solutions in clinical pathology that TechSite has developed or will be releasing in the next months and years to come, currently in research and development. And so we're excited to share more about that with you today. Our mission as part of that is to be the leading digital diagnostics platform for clinical pathology samples, right? We believe we are among the best in the world at helping labs analyze liquid and cellular and smearable sample types, digitizing them, using AI to be more efficient in making the diagnostic call. And we're to do that both in human, of course, that we're focused on today, and the veterinary world with animals and the environment. When we talk about our deployments in veterinary, TechSite has deployed in over 5,000 clinics with Pfizer Animal Health. now known as publicly traded Zoetis. Zoetis has now completed over 2 million scans with these veterinarian clinics among all these different applications, from fecal and blood to cytology and dermatology, for various animals, including dogs and cats and equine, and a lot more solutions to come. We are now over 2 million scans on that platform. And so our software really is robust and capable to scale in a very big way, in a global way. In the human space, this past March at USCAP, some of you may have seen, we announced our collaboration with Mayo Clinic. Mayo and TechSite will move forward with TechSite as Mayo's clinical pathology AI platform. And through collaboration, TechSite will be able to get more tests out to the world because of the expertise and samples provided by Mayo Clinic. So we're really excited about that. This will allow us to get more tests out to the world more efficiently and help all labs to be more accurate and efficient.
Now, as we begin the discussion today about digitizing and automating your lab, it merits a small recap of what led us here and the need for it. The first is an increasing demand. As the world population grows, the need for more tests keeps increasing. Increasing competition between laboratories, whether it be lab against lab or just lab against the nomenclature structures in place, make it ever more important for labs to turn negative contribution margin tests into positive contribution margin tests. Quality assurance. While the need for labs to increase their throughput is a very present element, the quality aspect is there as well, whereas training, certification, and quality assurance are essential. And microscopy, being a 400-year-old technology, is tedious and it's difficult, and it's very prone to repetitive stress injuries. And it is also associated with low job satisfaction. One of the ways that we like to illustrate this is an average slide that a lab tech can view during a day where they have to find Giardia. This is what they're looking at, and this is what they're faced. It's finding needles in a haystack. In this particular case, it happens to be there, but it's hard work. And the last challenge is the universal worldwide labor shortage. Laboratories are finding less and less candidates coming into the field, wanting to go into microbiology and microscope-based fields within the lab. And so there's increased need to find other solutions that help fill that void. In order to do that, what do? We turn to digitization and automation. And as Jacob alluded to, we will be going over the three major steps: digital slide management, reading, and computer-aided slide reading. Now, digital slide management is the first step. It's like the precursor. It involves things like barcoding your vials and slides rather than labeling them by hand. It's digital reports rather than paper, and laboratory information systems. And by and large, many labs have already implemented this step, which goes hand in hand with laboratory automation. The next step is digital slide reading, which starts with some potential adaptations to the quality requirements for specimen preparation. Now, this might require using different hardware, for example, avoiding incompatibility of equipment between staining or scanning racks, or other chemicals and consumables to facilitate subsequent scanning. And these changes can also lead to adaptations in personnel, cost of consumables, and processing time per slide. As well, the scanners that are used in digital slide scanning are a very important topic. There is no one scanner today that is the silver bullet. And so discussions around capacity, whether the scanner has sufficient volume, not just average daily, weekly, monthly, or yearly, but does your lab workflow have peak times that need to be accounted for? And then your workflow, do you prefer to have your samples run overnight, ready for the next day's morning samples, or do you need them to be processed throughout the day? Next, scanners don't have the same resolution. They don't have the same focusing or scanning speeds. The scanning area for your particular sample, is it a whole slide? Is it a portion of the slide? And making sure that you differentiate between overall scanner resolution and the numerical aperture. are all considerations that needed to be discussed. And lastly, file export. Does your scanner use a codec that's going to be compatible and optimizing lossy versus lossless for the AI to work on, or for the reads to be able to be done efficiently? And when you're planning your scanning purchase, maintenance repairs, as well as OPEX versus CAPEX, way of structuring those contracts, and availability and downtime are important considerations as well. Now, there are many scanner manufacturers on the market. We work with several of them. These are a few that each have their own positives and negatives, but it's a discussion that really does need to be had at the beginning, because it has a heavy impact on the whole rest of the process. Some of the environmental considerations that need to be handled are your IT infrastructure. It generates a lot of data, both in-house and sending out to cloud services. Some of the influencing factors are your sample size, the resolution of the images. Do you have Z-stacking? Is it bright field, fluorescence? What are the codecs that you're using? Are you sending out to the cloud, or are you doing it on site? And security. Now, for the workstations, generally the stations that are already in existence are fine, but 2 displays are nice with good graphics cards and dedicated input devices. And you want a large enough scanner, or a large enough screen with high enough resolution, but not so high that you're really having to move your mouse around and spending a lot of time getting from element to element. And then internet access is also one of the considerations, because you need sufficient bandwidth to be able to not have lags or stalled screens as you're viewing in laboratories. Now, the reading of digital slides before we get into any automation is very beneficial, because it allows you to get a better overview of your workload rather than physical slides. It allows you to manage your workload more efficiently. It enables remote consultation, curbside consultations, better interaction between colleagues. But the validation studies that are required before implementing digital reads are super important, because you need to make sure that, of course, your results are not non-inferior to whatever's in place. And you need to make sure that these tests are conducted in such a way that liability and regulations are respected. For example, in Europe, we are CE marked today, but we're for research use only in the US, pending FDA approval, which are underway. And then, of course, quality control is essential to avoid costly rescans during operations. Now, the third area that we wanted to discuss, and really the heart of today's discussion, is computer-aided slide reading. And as we begin this discussion, there are many technologies in labs today that have come forward, whether it be genetics, genomics, microfluidics. Today, we want to discuss artificial intelligence and focus in on that. Now, in the field of artificial intelligence, historically, the concept itself was actually defended by the polymath Alan Turing in the 1950s, who suggested that humans use available information as well as reason in order to solve problems and make decisions, so why can't computers do the same thing? But back then, there was a lack of computational power, there was a lack of data storage, and we didn't have enough data. Well, today we've overcome all 3 of those, and we have sufficient computational power, data storage, and plenty of data to be able to form these algorithms. Now, talking about algorithms, we are in the business of artificial intelligence, and more particularly in deep machine learning, which is the part of AI which gives computers the ability to learn without line by line telling them what to do. And within that, we use convolutional neural networks. I won't geek out on that, but it suffices to say that it's a type of machine learning process that uses interconnected nodes or neurons in a layered structure that resemble the human brain. Now, the result of that is that AI plus humans result in more accuracy, more efficiency, and time saved. In the clinical laboratory today, there are several tests that could be automated that are done through microscopes and that offer a very rich panel of possibilities for us to be able to automate.
Thanks, Troy. As Troy outlined some of these steps, we thought it would be helpful to share with you some of the computer-aided solutions that TechSite offers today. Of course, the title of today's webinar around automating and digitizing clinical pathology sample types, you heard Troy talk a little bit about scanners, and some scanners are the best in the world at scanning tissue or histology or AP, while of course TechSite has found so many differences around liquid and cellular sample types. And so we'll outline how these different samples, we've developed AI and workflow to help a lab be more efficient for these samples that quite often are much trickier to scan than a traditional tissue slide. The first is our product that we co-developed with ARUP Laboratories and that has been in production at ARUP for 4 years this month. The ova and parasite detection tests that we offer, we have one for trichrome and for modified acid fast. We have a wet mount test being released here just in the next couple of months. But these methods, those of you in the parasitology world, you know that there's traditional microscopy, there is PCR, molecular, to identify certain parasites, and there's also the antigen. And you understand probably the pluses and minuses of each of these various methods. The gold standard generally remains as microscopy. Our AI and workflow for O&P includes a classifier and counter that identifies these variants, these parasites, both for trichrome and MAF. And all of the ones you can envision that you want for a wet mount test when we release that, those will be available as well. But we are helping the technologists and pathologists, parasitologists, to take their manual review time that's traditionally 4 to 6 minutes under the scope, looking for in the presence of one parasite like that Giardia that Troy showed you earlier, and cutting that review time from the 4 to 6 minutes down to, on average, 20 to 30 seconds. So those efficiency gains in an area where there is a declining skill set, many of you have probably shipped your O&Ps out to reference labs because you haven't had enough of the skilled labor for this particular test. The O&P solution from TechSite can certainly help, particularly as more folks are retiring in this field and not as many are rising up. I mentioned that we've been in production about 4 years now at ARUP with the test. There are other laboratories using it in production today as well. There is a couple of articles in JCM in the Journal of Clinical Microbiology about our ova and parasite test, including an abstract commentary. Feel free to read more on that. And I do want to make clear that these slides and the recording of the webinar will be made available to all of you attendees as well. So don't worry about capturing images. You'll be able to access them afterward easily. From a regulatory standpoint, you heard Troy mention that we are RUO and we are CE marked for several of our solutions and our platform in Europe. In the United States, we are RUO, research use only, but we are on a path to regulatory, various spots in that path for each of our different solutions. And so that is true about all of ours, but they're all on the path to FDA. Troy.
Thanks. Now, our first collaboration with Mayo Clinic is the Bacteriology Digital Manual Review. Now, this particular solution came about because Mayo Clinic saw the value and the need in having remote reads made possible through gram stains. Now, the current process is you've got your gram stain review, you do your culture workup, you've got a collaborative review, and then you have to manage slide storage. But the microscope is difficult, slide disposal is messy, storage space is an issue, and then retrieving and locating and rereading slides is not ideal. Now, with our solution, we are able in one interface to allow any of the bacteriology gram stain reads from all of these sample origins that you see on the screen to be read at low power and then high power with the normal digital navigation, annotation functions that you would expect to be able to do in manual reads. And that's exciting because it allows for the remote reads, but it also sets the stage for what's coming because we are currently under development for the AI component of all of those sample origins as well. Now, that came about because we had already developed our bacterial vaginosis gram analysis, which today you've got your culture plates that start growing, but then you need to challenge or confirm what's on your cultures with the direct exam or the gram stain, because the patient, as they come into their doctor, might already be on antibiotics, and others simply don't show up well through culture plates. And so the gram analysis is an essential component of BV detection. Our solution today is AI powered and enables for detection of all of these organisms. And we also, through this, have completed our technical study, which revealed that in terms of sensitivity, we're at 96 to 100%, but specificity was a bit lower. And the reason it was a bit lower is because our solution was able to detect more transitional or type 3 Hayisen, which are positive BV cases. So it's actually a good story for us to tell. We're currently bringing this to market as well. It's CE marked, and we're going to be continuing with the other sample origins shortly.
We also have in the CP space a automated blood differential solution for those traditionally doing a manual differential. A white blood cell, red blood cell digital differential is offered by TechSite. We are scanning the whole scan, the whole slide, and allowing users to go and look at the whole blood smear. We are also doing a digital differential. The AI is pre-classifying and giving counts and percentages. It's doing the differential. Like all of our tests, a user could change or confirm any of the pre-classifications. We also, our model for all of these tests, I should make clear, the number of users within a lab or outside a lab is infinite. We charge per scan that is scanned into the platform. And so your ability to collaborate with colleagues is not inhibited. If you are scanning and getting these solutions provided by TechSite in the web browser, cloud-based, you can collaborate and share. Our blood differential has been published in ISLH around its sensitivity and specificity. And this one is the most advanced in our regulatory pathway. This one, we submitted our 510K this past spring, and we are working through that process now. We also offer a solution for cervical cancer screening, a pap smear, cervical cytology. Our algorithm is robust and supports both liquid-based prep types, thin prep and SurePap. So some of you might already be digital with one of those preps or LBCs, but not the other, and still have manual microscopy. But we use the Bethesda classification system, and our algorithm would handle both of those liquid-based prep types. This is in validation in multiple labs globally today. And our UI is really designed to help a cytotechnologist, a cytopathologist be more efficient, just like all of our tests. The atypical cells are pre-classified by TechSite, and then the user can do a sub-classification. Efficiency gains are significant here as well, greater than 80% time labor savings. You're able to still go into the actual whole smear and go look if you'd like, but generally the users are able to stay in the images view on the left-hand side and identify the pre-classifications and confirm their diagnosis. Regulatory pathway as well with this solution. We also have a novel bladder cancer screening solution. This is not a traditional microscopy-based test, but a neat opportunity for laboratories to be cutting edge. Traditionally, bladder cancer in the US alone has 1 million bladder cancer survivors annually or so. And you can see the recurrence rate is high. It is among the leading cancers for men, among men. And the current processes, the current methods vary, and they aren't overly sensitive. So today, the standard is a biopsy through the urethra to recognize if there's a recurrence of bladder cancer. And so a pre-screening tool that's less invasive could be helpful. We have partnered with a company called Cytobay. Cytobay has developed a proprietary stain in urine. Bladder cancer, urine samples can actually reflect the presence of bladder cancer cells. And Cytobay has developed that stain, and using our AI platform, TechSite is able to quickly identify if there is recurrence of cancerous bladder cells, which is a nice pre-screening just by peeing in a cup rather than an invasive biopsy. And so we've been excited about the labs that have wanted to try this out and demonstrate that there is a new, less invasive approach that allows to identify the presence of ICC positive recurrence of bladder cancer cells. And Troy will share a bit about our digital fish solution for hematopathology.
Yeah, this is actually our first foray into non-bright field microscopy tests through the use of fluorescence. And our digital fish applications, in addition to hematopathology, are going to cover pre and postnatal diagnostics, GTC, mosaic karyotyping, and fertility studies. Now, what's exciting about this is that it's the same workflow as the others, and the cell images are uploaded to our platform. We then perform a signal pattern pre-classification, whereas then the technologist then is able to quickly either confirm or challenge what the AI shows and compile the report, which is sent to the LIS. And the result is, for this test, the increased lab efficiency, side-by-side comparisons of digitized cells, which of course improves accuracy, classifying and rejecting the marked patterns and cells becomes way faster, and improved identification of chromosomal aberrations. And so we're excited for this solution to be able to come to market as it opens up yet another realm for us in our diagnostic space.
So you've heard a bit about the clinical pathology AI platform from TechSite. The capacity of the platform to analyze liquid and cell and smearable sample types is vast. We have a long runway. We are excited about developing and adding more tests in collaboration with Mayo. Among those products coming and about to come, we do have a super resolution product, for example, that will, using software from TechSite proprietary, allows a image that was scanned at 40x to become 100x, just via some technology behind the platform. As one example, you can see the applications from less expensive scanners at a lower resolution, taking those images, and TechSite able to enhance them, allows more economy of scale. Even laboratories that might not have the capital expenditure budget could start to approach some samples that historically would require a much higher resolution. Tests that we currently offer, you've heard them outlined today, right? Our current solutions in CP. There on the bottom, you can see our product roadmap. We are headed to co-develop these tests with Mayo, their data, their samples and expertise, along with TechSite's world-class platform to iterate and develop algorithms among the best in the world and take them out into laboratories for production use. You can see the solutions, additional ones in blood, additional ones in microbiology, additional fish solutions, even moving into the mycology space. We have our SporeSite solution for environmental that we look forward to easily transitioning over to the human space. So our platform is excellent at ingesting these unique types of samples that quite often historically have not been able to be scanned well. We figured out ways to scan them, and our algorithms, our data management and engineering teams are building really strong algorithms and models that have been collaborated with and verified by experts like Mayo and ARUP to help laboratories like yourself to drive efficiencies and help with tests that might be losing money today, to help you to start to make money with them. One other one that I'll end with that's pretty cool. We all know sepsis is such a tragic challenge in all over the world, especially in the United States today. If there was a way to detect that faster, and we believe that it's on our roadmap. We're talking with Mayo around co-developing that here in the near future. We believe using our platform, we've got the capacity to help laboratories and hospitals identify sepsis even earlier. So just share these as examples of what TechSite, the clinical pathology AI platform, can do and how it can help you to automate and digitize and ultimately improve patient care.
Thanks, Jacob. One of the other considerations, aside from the tests that are actually available today that can be automated in the lab using AI, is the process and change management. From a technological standpoint, digitization in general and automation through AI in particular come with an increasing complexity and the number of interacting hardware and software components. This, of course, increases the potential for downtimes of single components, which affect the entire workflow. And these issues can be mitigated by planning for sufficient redundancy to meet the uptime goals on your estimated downtimes. From an administrative standpoint, digitization of a lab involves procurement and installation efforts. Details have to be negotiated, contracts signed, hardware and software delivered, and systems need to be installed. From an operations standpoint, as workflows change for all lab personnel, training is of course needed for everyone involved, and the workflow is designed to accommodate the new technology. And then moving forward, the personnel need to be certified and or recertified over time. From a personnel level, buy-in and adoption of any new technology by the personnel is critical. A good example of this, actually, Jacob spoke of earlier. In 2019, we launched our fecal in production at ARUP, which is one of the largest reference laboratories in the US. And through the project's implementation and putting it into production, we quickly learned that the personnel absolutely loved the new technology, didn't find it a threat at all, and in fact, embraced it immediately. And so by making sure that we were there for them and accompanying them through the process of not just implementation, but afterwards training, we were able to turn that into a vast success for that company. The last element that we wanted to cover today are the economic and performance metrics. Now, there are two main ways of measuring them. The first is through turnaround time, and different requirements and equipment may increase or decrease the convenience and processing time of the individual steps, which impacts the total turnaround time, which is likely to decrease if the time savings coming from the transmission of the digital data faster than shipping or couriering physical slides, for example, is less than the manual work that is introduced in the scanning. The second consideration are the costs, and these can often involve additional investments and or savings. You have one-time setup costs, which are like installation and training. You've got periodically occurring costs, which are like scheduled maintenance, replacing hardware after their end of life, and continuous costs, which are like shifts in personnel, consumables, or per test costs. Now, there are two challenges in this that need to be addressed. The first are that additional costs are often easier to calculate than the financial gains. And what that means is the financial gains often don't just revolve around reduced time, but moving into the second challenge, they often are related to not easily convertible to monetary amounts aspects, meaning job satisfaction, for example, or ongoing health or attractiveness to employees in a competitive job market. In particular, for innovative faculty and staff, it's not just a matter of salary. Now, throughout all these considerations, I just want to reiterate one of the slides that Jacob presented in the beginning, in that TechSite as a company, we are an AI company, but the AI is just one component of the expertise that are required to roll out digitization and automate a clinical lab. And the elements that you're seeing now, from, of course, the AI and the data curation, to the image management system, the platform that's behind it, the image acquisition, the custom workflows, the security, the scalability, and of course the integration with LIS. Those are all expertise that are critical into automating a lab. And ultimately, whether it be improved efficiency, the faster turnaround time, the increased accuracy and precision, but also the resource optimization, the improved decision-making capabilities, and continuous learning and improvement. One thing is for sure, it's that the clinical laboratory as of today will never be the same with the advent of AI. And our hopes as a company, as TechSite, is that we will be able to accompany anyone throughout these transformations in their clinical laboratory.
Thanks, Troy, and thank you all for your time today. We have had some questions come in, and as you have additional questions now, please continue to enter those in the chat. We had a question from Mark. Can we share results with colleagues? Is there a cost per user? How many users are allowed? Great question, Mark. Thank you. We do not charge an annual licensure or fee. Our model is per scan that is uploaded to TechSite and analyzed by TechSite's AI with the results presented in the web browser to the laboratory. That's the charge. And so the number of users is unlimited. Certainly the shareability to other users within TechSite or even non-TechSite users to get a view-only link, that's all unlimited. Thank you. Troy, looks like we've got...
Yeah, I can handle Richard's question. How does this compare with sensitivity of the pathologist or the human's review? Every single one of our tests starts off with, in fact, as a company, we are ISO certified in that we follow a very strict set of criteria even before we start a project. And one of those first criteria is establishing the key performance indicators of our algorithms. Now, these come from the gold standard existing technology or processes for every one of our tests. We first establish that as a benchmark, and then we model our sensitivity and specificity objectives according to those. We then validate those through a complete battery of testing that ends with clinical trials and technical validations. So, short answer to the question is that we are always at least at the level of a human review, but the reality is that so far in all of our tests and trials that we have performed, we perform better.
And those data points are reflected in many of the publications out there, which are also easily accessible at techsite.com. Thank you so much. Great question. And Troy, appreciate it. I learned some things too. Janet asked, how long are these results stored in TechSite? So the standard offering of the charge per scan, that includes 30 days storage of the image counts, classifications, any of that data would be hosted in the cloud for that long. Certainly possible to keep it there longer, but the standard model would be 30 days. Most laboratories find it more cost effective to import those results to their LIS and save locally or in another method that they're typically preserving patient results in.
I'm seeing a question here from Don, who asks about the costs for evaluating these kinds of tools. And that is entirely possible. Our standard process is to consult with interested labs, because as we've discussed today, there are a good number of discussions that are very particular to every lab setup. And that enables us to get both a scope in projects and be able to make concrete recommendations, as well as try out our system even live before any costs are incurred. So that's entirely possible, yes.
And let's see, Yun Zhong asked, you'll update the AI function if it's AI based. How do we get access to it? So, Troy, I believe he's probably asking, how often would we update or release an improved algorithm?
Yeah, in fact, it's a good question, because when we first launched TechSite, we were very idealistic and loved the fact that our system actually enables real-time feedback and real-time model updating, in that we can ingest every feedback that our users give our systems as they run through their daily workflows and incorporate those into new models. Now, the reality is quite different. And the reality revolves around regulation. The paradigms that the FDA currently works under don't really know how to handle something like AI. Now, our relationship with them has been great. They've been great to work with, but there is a pretty large gap between the way that the in vitro medical device regulation is set up, where you got a product, you have to freeze it in time, and then you release it. And so currently, as Jacob alluded to, we have release schedules where we do update our models, we do improve them, but it's not real time, because while we are in new technology, we are existing in a traditional medical environment where you need standards and you need to follow regulations, and we're very dedicated to that. So we follow the guidelines by the FDA, NCE, and we manage a release schedule with the updated AI. This being said, none of our products come to the market to begin with before they already are at or surpass their human counterparts. And so any updates to the AI that come after is just kind of a cherry on top of the cake, where it just keeps getting better.
And only would release for clinical production use when a laboratory is ready to upgrade to the newest model. Yeah.
Yeah, sure.
Stephen asked, how would this work with reporting in my LIS, in my laboratory information system? Excellent question. So there would be two ways this could work. We have several customers that have the TechSite web browser open on one side of their monitor or on a monitor, and then they have next to it open as well their LIS. And so they might choose to manually enter results from TechSite, and those who are looking to get efficiency gains quickly normally do that and eventually work with their IT teams on their side to integrate their LIS with TechSite's fully open API, application program interface. TechSite has that open API, which will allow a laboratory to integrate the LIS, and that, of course, adds another efficiency when images, data, classifications, counts, diagnostic result, all of that could be imported automatically from TechSite into the LIS, further improving efficiency. So both would work. We see most labs, because IT takes a long time, and that's just a general rule of thumb. They like using TechSite immediately, manually entering the results, but eventually getting to the point where the LIS is integrated, so that part is no longer manual. Anything you would add there, Troy?
No, we do have the API, and it does work with any LIS. Yep. And we have a question from Mark, who, it's actually a good question, what labs are using TechSite? Many of our clients are under NDA. However, some of our more public ones, where we've been able to announce collaborations, such as Mayo Clinic and ARUP, are already using our systems. And what I would encourage is for you to just reach out for us, and no obligation, we'd love to share with you some of our other implementations on our technology on who is using our systems. And if I can just touch on this one as well, Rebecca asked, what scanners do we recommend? This goes back to the slide where there is no one scanner. There are so many issues from the scanner's capacity to its specifications, how it scans, the resolution, the numerical aperture, how it exports files, that we need to go through with each individual installation before we can recommend a scanner, to where it's impossible to just blindly recommend a scanner. Because depending on what you want to do with it, your lab's throughput needs, the workflow, and all the other considerations, of course, it varies.
Piggybacking on the scanner question, there was another question here from Mark again. How much do digital pathology tools like this cost? So, of course, the scanner would be one expenditure. And some labs make that a capital expenditure. Others turn it into an operating expense, and we can work with our scanner partners to help you get it how you would like for budget purposes. But for the ongoing software use from TechSite per scan, the pricing is such that it fits within existing CPT reimbursement codes. And depending on annual volumes, most of the solutions you saw today will reimburse in the range of, the charge per scan would be between $2.50 and $5.50, depending on annual volumes of sliding scale. So most of the solutions today are in that range, fitting within the existing CPT. We are finding some labs are getting so much labor savings time in their own current metrics that they can turn tests that are currently losing money to actually being very profitable with this model. I'm not seeing any other questions come in. Really appreciate the engagement. Your questions were spot on. Thank you so much for your involvement and for listening in today. We appreciate your interest in learning about digitizing and automating clinical pathology in your lab, and of course, learning more about TechSite today. Troy and I appreciate your patience with us and your time. May you have a great Tuesday. Thanks again, all.
Bye-bye.